want to make a small announcement, and that is, you guys got to be here at 9:30, because I'm getting my ass out of bed early, and I'm here at 9:30, and I want to get this. I want the talks to start when they're supposed to start. Otherwise, we should just set the time for 10, Paul. There's no point in having a 9:30 start time.
No, but it'll start at 10:30.
Yeah, well, I know, that's the problem, but really, I got here at 9:30 today, and there were about 5 students. But all the speakers were here. I'm very proud of you speakers. You're very good. So yeah, get here on time. I think that shouldn't be too hard. Okay, so our next speaker is Steve Nocter. He's at the Mind Institute at the Center and at UC Davis. And Steve is going to talk today about evolution and regulation of neural precursor cells in the developing vertebrate brain. And what I really like about Steve, so I would say the vast majority of people who work on cortical development work on a mouse. And the molecular mechanisms involved in expanding the cortical sheet, cell cycle kinetics during development, aerialization of cortical fields, they work on a mouse. And a mouse has kind of a pretty shitty brain. And it's only one animal, so how that changes is hard to know. Steve is one of the few developmental neurobiologists who actually uses a comparative approach. And he not only uses a comparative approach, I don't know if you have read his work, but I highly recommend it. He does really, really beautiful studies, robust, and his papers, some of his papers are a tour de force. That paper in PLOS was just extraordinary. It was a comparative paper. So I know you're going to hear a really great talk today, but he, like I said, he does think about evolution, and he uses a comparative approach, and not a lot of people do that. So I'm really glad you're here. I don't have a funny story about him, but give me a couple days.
For the invite. I'd like to thank the organizers for allowing me the chance to share my thoughts with you. This has really been a wonderful meeting. It's been very well run and a fantastic location. So with that, I'll move on. And what we're going to talk about today is precursor cells in the prenatal brain. So what's going on in the prenatal brain before development? What you're looking at here is a coronal section of macaque brain. And down here in the lower right, let's turn this on. Down here in the lower left, you've got a proliferative zone, and then emanating from that proliferative zone are these twirling fibers. These are fibers that emanate from the precursor cells, and they help guide the migration of cortical neurons. This is a higher power image of that. And this really beautiful, interesting pattern is something that you only see in primates. You don't see this in rodents. It's really wonderful. So now we'll be talking about cerebral cortex, and of course that is this outer gray layer here that sits on the surface of the bumpy brain underneath our skulls. This is what it looks like in the adult. We won't be talking about the adult, of course. But just keep in mind that according to people who count the numbers of cells, we've got roughly 60 billion of them there. Around 20 billion of them are neurons. The other 40 billion are glia. This is work that's coming from the Packenberg lab in Denmark. These are mostly generated during development. When I was growing up as a student, they taught us that everything was produced by 20 weeks of gestation, and that's just simply not right. And as you've learned over the past couple of days, development of the brain can be influenced by many factors, just something to keep in mind. Now, I like to share a little bit of information about myself so you can get to know me a little bit better. This is the earliest picture I could find of myself. This is my mom sitting down to dinner. We had dinner at 5 o'clock every night. A bunch of brothers and sisters. You might be wondering which one is me. You know, you got this talkative guy here, that's not me. The hungry one, not me. The thoughtful one here, not me. If you're wondering...
Took the picture.
Huh? Yeah.
Technically adept.
Precocious photographer. Now, in fact, that's me right there. So this picture was taken when my brain was developing. And as we've learned, you know, the stress that my mother probably endured by having these 5 children already was probably not good for me. But, you know, nonetheless, I'm here. She's not smoking, no.
That's not an excuse. Come on, man.
Yeah.
That's great.
Yep. But here I am. So these cells are mostly generated during development. And one thing that I want to point out to you is that in the brain, in the adult brain, you've got this cavity, the ventricular cavity. This is the lateral ventricle. You've got paired ventricles, 1 and 2. You've got a third ventricle down here. And this is something that will play a role or that you should note. Now, over there on the far right, I've got a human brain at gestation week 11, and these are at the same scale. And interestingly enough, by the 11th week of gestation, the human brain is the same diameter as a rat brain, and it looks kind of similar. So if you've looked at sections of the rat brain, it's about the same. So by 11 weeks old, we're little rats. But again, you can see that little cavity right there. And the reason that I note that is that there's a proliferative zone, that proliferative zone of all the precursor cells, and that lines the cavity for reasons that we don't fully understand yet. Another thing to point out, and this is a drawing from Rakesh, is that here's your cavity again in the middle, and there's this radial deployment of precursor cell fibers that emanate in a radial direction, kind of like spokes of a bicycle wheel, away from the cavity towards the peel surface of the brain. So this is in the occipital lobe. And another point that I'd like to make is that characteristics of the brain change pretty dramatically from one region to the next. So here you're looking at a gestation day 80 macaque brain, and you can see these precursor cell fibers. In the macaque, they're labeled with GFAP, and you can see them kind of emanating away in a radial fashion, but they're clustered in these fascicles. And this is in same stage, but in somatosensory system. And this is the medial wall. Again, you can see them kind of emanating away. And this is in the dorsal cortex, and I don't know if you can get a flavor for it, but the trajectory that these fibers take is not always radial. It really depends on the stage of development and the area of the cortex. So this is, again, that high power figure, and you can see that you have these fascicles of these twisted bundles of precursor cell fibers. So the point that I want to make, and this is true of many things, is that these facets that we'll be talking about that occur in development, they are very much dependent on place and time. So back to me. So I was born in New Jersey, and I grew up in New Jersey, spent a lot of my education, had all my education there, and then ended up in California. And as you can see, California is quite a bit larger. So this is at the same scale again. So, you know, being an anatomist, I like to keep things to scale. So New Jersey is much smaller. And in fact, when I moved out to California, I found out that everything's bigger. So you know that you have these giant redwood trees in California, and you can go visit them. They're quite amazing. And here I've got 2 pine cones, and one of them is the pine cone for a redwood tree. Anybody want to guess which one? It's the small one, yeah. So that's the redwood tree, pine cone. So there it is. It's about the size of a penny. But if you have a green hand or green thumbs, you can plant them, and they will grow, and they grow very rapidly. And in fact, this is outside of San Francisco, if you ever happen to visit. And these are young trees. These are only about 800 years old, these ones. But the largest ones are much taller than many known landmarks. In fact, the tallest tree that was recently identified, it's taller than this one now. So the tallest tree used to be 379. They found another one that's close to 400 feet. And they won't tell anybody where it is because probably there's some fool out there that would try to cut it down or do something crazy to it. So they're quite tall. What is the point of all this? The point is that great things can come in small packages. And that same idea is true with the nervous system. So what you're looking at here is the developing nervous system at 3 weeks of gestation, just after the neural tube closes. So the nervous system starts out as a flat sheet, it curls up and it closes on itself. And then this becomes your ventricular cavity. So that's the central nervous system at 3 weeks of gestation, and it's teeny. So it's 7/1000ths of an inch, 100 microns, 200 microns thick. But that teeny little structure has all the instructions, and it has enough cells to make this very complex organ. So to kind of keep things in perspective, that little dot there, that's your nervous system at 3 weeks of gestation. In fact, it's even smaller than that, but that was the smallest that PowerPoint would let me make it. So there's your brain at 3 weeks of gestation. Here it is again, the rat brain at 11 weeks of gestation. There's a fairly steep growth curve. So we've got our 60 billion cells. They're generated during development. And if we look at our brain and then zoom in a little bit more, this is a section of tissue that's stained for cells. So regions that have lots of cells stained darkly. And you can see that there's a very dark band around that lateral ventricle. Those are the precursor cells. They generate your neurons and glia, and they migrate out to set up shop in the cortical plate. And so that band out there will end up becoming the gray matter that sits on the surface of our brain. So that's the cortical plate. Now, the Packenberg lab did this really interesting study where they counted the total number of cells in the cortical plate at gestation week 13 and again at gestation week 20. And as I told you, we had been taught, and you can still find it cited in some places, that everything's in place by, or has been generated by gestation week 20. And what they found is that in 7 weeks, the total number of cells grows by 5 billion. It starts at 1 billion, ends up at 6 billion. So at 20 weeks, you have 6 billion cells. You have to get to 60 billion. There's a lot that's going to happen in the second half of gestation. But here's what's kind of interesting. 5 billion cells are added in 7 weeks. If you do the math, what that ends up being is over 1,000 cells a second. So 1,000 cells. 1,000 cells. It's actually 1,184, but that's harder to say. 1,184. 1,184. It's a lot of cells. So, and keep in mind that there's two basic cell types. You have excitatory, whoops, jumping ahead of myself, excitatory projection neurons and inhibitory interneurons. I'm going to be focusing almost exclusively on the excitatory cells. And they're all generated from this proliferative neuroepithelium that lines the ventricle. The excitatory cells have a shorter distance to migrate. So they're generated and they migrate out radially. The inhibitory neurons, at least in the rodents, they're generated primarily in a structure called the medial ganglionic eminence, and they take a much longer tangential route. So again, I like to put these things in perspective. So this is a relatively short distance, but the brain gets larger and larger and larger in development. And in human, the comparative task is if I was one of those newborn neurons, and to consider the distance I would have to traverse in order to get where I'm supposed to go, you would stand me at the base of the Empire State Building and I'd be looking up at not just one, but you'd have to stack 4 of them on top of each other. I'd have to climb that distance and get to the right place. So it's really a remarkable task that it happens well enough and often enough that we can sit here and consider it.
But now, so we're really only talking neurons here, or this is mixed neurons and glia?
That's neurons.
Okay.
Yeah, the glial cells, I don't think we understand gliogenesis well enough yet, honestly. So given the vast amounts of proliferation, it's not surprising that sometimes things go wrong. And this is an example where things have gone drastically wrong. This is lissencephaly. You can see a normal brain or a control brain on the left, and on the right, the lissencephalic brain, greatly reduced in complexity and in the number of cells, and it's much, much thinner. And these cortical malformations, there's a variety of them. They can result in problems with dyslexia, learning abilities, epilepsy, mental retardation, and they can result from deficits in migration and proliferation. This has a huge impact on society. So if you consider epilepsy alone, in the United States, there's about 2.5 million people each year that are affected. You look at the cost, it's estimated at maybe $12.5 billion. Another neurodevelopmental disorder, schizophrenia, perhaps 2 million people that are affected every year, estimated cost to society $62 billion. Autism, 1 in 88, more recent studies put that figure even higher, 1 in 68. And one estimate that I read in 2007, 35 billion. And keep in mind that those are purely medical costs, medical treatment. They also include loss of work time, but it doesn't really in any way account for the difficulties faced by these people who are affected or the families that have to deal with these people. It really comes at a tremendous cost, and that's why we work on this stuff. So as Lee was mentioning, most research involves animal models. Here's our mouse. I don't know what % of the work is on mouse, but it's a great deal of it. Another model that I've worked on a lot is the rat. The rat brain is bigger. It has 3 times as many neurons as the mouse brain, according to some stereological estimates. The ferret brain, this is another model that I was fortunate enough to work on, and you can see the ferret brain is bigger than the rat brain, but it also has these gyri and sulci, so it's a more complex looking brain. Now we're moving on, we're working on primates. We're lucky enough to be at UC Davis where you have a large primate colony. And then of course, what this is all getting at is trying to understand the human brain. And so how relevant are these models? You look at all this work done in the mouse, does that help us understand the human brain? And to some degrees, it does. And one thing that is notable is that if you look at a single radial section of the brain in rodents and primates, they are composed of the same laminar structures, the same types of neurons in all the lamina. The thickness in a rodent would be from 0.8 to 1.5 millimeters. and the primate brain can be much thicker, 1 to 4 millimeters. But where the biggest difference is in surface area. So rodents have a surface area of 6 square centimeters, human 2,500 square centimeters. So we have a lot more processing power. Now, you're probably aware of this, but if you look at the cortical sheet, you can see that there are different cell types. And in low power pictures of some cortical areas, it really jumps out at you. You can see all these bands. So these bands are comprised of different cell types. And they're generated, as was mentioned previously yesterday, that there's an inside-out generation of the cell. So this is from a 1974 publication by Pashka Rakesh. He's injecting the tritiated thymidine at different dates, so embryonic or gestation day 45 up to 102. This is in a rhesus macaque that has a gestation of about 165 days. So basically, around the middle third of gestation. And early injections will label deep layer neurons, and as you get later, you get progressively more superficial neurons. I did the same thing in ferret model. We worked in somatosensory systems, so here's a section through the ferret brain, and we looked at a very specific region and injected BRDU, for example, at embryonic day 22 out of 41 in the ferret, and you would label interstitial cells in the deep white matter. They start out as subplate cells. They don't actually disappear. They remain there. E26, you have cells that are subplate and deep layer 6, 28, 30. So you get the point. So you have this inside-out generation, and I'll explain why that may be important later on in the talk. So in my field, one of the questions that drives a lot of people is how is cortical expansion achieved? And the vast majority of the work is considering what are the mechanisms that promote growth. People ask questions, how do you make a bigger brain? And in order to understand that, I think we need to have a firm grasp on what are the types of precursor cells. And we're working on this work. I think it's good for us to always keep in mind that we're not the first to do this, that people have been working on these questions for a long time. And in fact, what you're looking at here, this is work from over 100 years ago by Ramon y Cajal. And Cajal was an artist. You may know that already, but this is one of his drawings of a rabbit brain. And he's labeling these precursor cells. So here they are near the ventricle, and they have these long, thin fibers that we've been talking about already, coursing up to the peel surface. One of his colleagues, Wilhelm His, also worked on it. He wasn't quite the artist. So, in fact, this is a cartoon, a drawing that Cajal prettied up and reprinted in one of his publications. But what His noted is that there are what he called germinal cells, dividing cells, and that these are located along the border of the ventricle. And what His concluded is that there were 2 cell types in that proliferative zone. There were mitotic germinal cells that they were doing the work, they were producing the neurons. And then there were these bipolar radial cells that he called spongioblasts, and that wonderful name stuck around for a while. So moving forward to some other notable findings, there was a work by Frederick Sauer that was published in 1935, so he worked in Wichita, Kansas. And what he did is he did something very simple, but oftentimes simple approaches are the most powerful. He was an expert in mitotic cells, and what he did is he looked at all the cells in the neuroepithelium, that proliferative zone that surrounds the ventricle, and he did this in pig, which is a gyroencephalic animal, and also in chick. And he would look at the nuclei, and he would put them in a class of division, and he divided up into about 10 different categories. And this would be cell type A, and the dividing would be cell type J. And he would look at every single cell, he would give it a class, and then he would measure the distance of that cell to the ventricle. And by using that approach, what he showed is all the intermitotic cells were far away from and the interphase cells were far away from the ventricle, and the germinal cells, or the dividing or M-phase cells, were at the ventricle. And what he concluded is that the germinal cell and the spongioblast were actually one and the same. There weren't 2 cell types, but it was the same cell. And what he proposed is that these cells, these neuroepithelial cells, were bipolar cells, and that they would all divide at the ventricle. And what that means is that they exhibited this characteristic that's called interkinetic nuclear migration. They would bob up and down in time with the phases of the cell cycle. So S phase, where they would synthesize DNA, would be at the top of that proliferative zone. In G2, they would drop down at the ventricle, and then G1, they wander back up. So he made this proposal, and then unfortunately, he passed away. His wife was working with him at the time in Kansas, and after he passed away, she was an anatomist. She decided to move away. She went to Texas, worked in a medical school in Galveston, and started working on vocal cords. 15 years pass, 20 years pass, and people start writing reviews saying, remember that stuff that Sauer came up with? Well, it was bullshit. It was all wrong. That can't possibly be. And she read enough of that, and she just got fed up, and she said, all right, I'm going to test it and figure out what's going on. And she used the most advanced techniques that were available at that time, which was the tritiated thymidine analogs. So she did this in chicken eggs. She would just inject the tritiated thymidine into the chicken egg. And the way that works, if you're not familiar with it, when the cell is synthesizing DNA, it'll incorporate the thymidine. And if you've injected tritiated thymidine, it's going to use that too. And then that gives you a tag, and you can find that tag. So she would inject the tritiated thymidine, wait, 15 minutes, and then sacrifice the animal. And she found that, lo and behold, in the nervous system, all of the labeled cells would be away from the ventricle. So that's where the S phase was occurring. And then she would inject the tritiated thymidine and wait progressively longer times. And as she did that, that cohort of cells would march down to the ventricle, and they would undergo M phase there. Progressively longer time points, they would start marching back up. And they would continue doing that. And so she proved that her husband was right, and she published not a paper, but an abstract at a meeting in 1959 saying that, well, damn it, he was right. so that cemented this idea that there's this interkinetic nuclear migration. So Sauer first proposed it in 1935, and people are working on it today, and what they're finding are some molecular motors that control that movement, but we still have no idea why it's happening. We could guess. There could be many good reasons. So today, we have some markers that allow us to identify mitotic cells, and you're looking at an embryonic day 17 rat brain that's been stained for cyto11, which labels the nuclei of all cells. So you can see each one of these green dots is a cell, counterstained with an antibody for M phase cells. So you can see that there's all these M phase cells undergoing division at the surface of the ventricle. But it turns out that's not the entire picture. There are some cells that are dividing away from the ventricle too. And those cells today, we call them components of the subventricular zone. And again, this is not something new. It's actually been known for quite some time, but some of these papers remained hidden. So Alice Hamilton published a paper in 1901 looking at the subventricular zone cells. She had a different name for them. She called them extraventricular. And what she did, again, was very powerful. So she would make sections of the brain, and using the appropriate stains, you could identify the dividing cells. And at the time she was doing research, there was this great divide. There were two camps. There was one group of researchers, and they said every single cell undergoes division at the ventricle. And there was another camp saying, well, actually, no, the majority of them are dividing away from the ventricle. What are you talking about? And what she did, she was the first person to actually do a developmental time study. So people who said everything's dividing at the ventricle were looking at very early stages of development, and they didn't think, well, things might change if I look across development. And other people were looking at late stages. She actually looked at the Hall gradient across development, and she concluded that over development, the proportion changes, and towards the end of the development, the majority of cells are in fact dividing in the subventricular zone. Early on, it's all at the ventricle. The subventricular zone comes into play later. So, because of the wonders of the internet, I actually stumbled across and was searching and found out a lot of interesting, this is a little minor aside, but she really has an interesting history. She wanted to go to medical school in the United States, and at that time, women in general were not allowed to. So she applied to many places, and many of them just said, no, women don't go to medical school, what are you talking about? But she managed to get into the University of Michigan because her family was well connected. And then she wanted to do postdoctoral studies, and she did research into where some interesting work was being done, and she wanted to study bacteriology, and she said all the best work is going on in Germany, so she applied to positions there, and again, she was told, no, women don't work in labs. But because her family was well connected, she managed to get into Carl Weigert's lab, and he said, you could work in my lab, but you have to make yourself inconspicuous. And I don't know what that means, maybe wear a baseball cap, put your hair up, I'm not sure. But she managed to do the work. She worked there for 2 years, and like most work, her projects didn't pan out. So she worked there for 2 years, returned to the US, and she started working on public health issues. She identified the causes of typhoid fever and tuberculosis. She demonstrated the dangers of lead exposure in the workplace. So she was working in the Chicago area. They had these factories where the workers were exposing themselves to lead. They would get sick, call in sick, and get fired because you can't call in sick back then. And they would hire new people, they would get sick. They were, these factories were just rolling through people. She was the one who demonstrated that this is not good for you. And so single-handedly, she founded the field of occupational health medicine, and Illinois founded a commission on occupational health, first in the world. And it was during this time that she conducted and published her story. This was in the Journal of Comparative Neurology about these subventricular zone studies. She was writing letters to her sister about how hard it was to get published, so nothing's changed. And then she became the first female faculty at Harvard. So Harvard said, wow, this occupational health medicine, this is interesting. We're going to start a department, but you're a woman. You're not going to be the chair of the department. In fact, we're going to maybe allow you to be the first female faculty. You can come here, but you can't go to the faculty club. You cannot go to football games, and you cannot go to any ceremonies. So again, she had to make herself inconspicuous. But she agreed to it. This was progress at the time. And finally, if you are this sort of person and you do that sort of work, you might get yourself a stamp. So she's on a stamp, noted as a social reformer, not as a neuroscientist, but she did a lot of other work. So really a remarkable person. So back to the story. So we have these cells that undergo division at the surface of away from the ventricle. So remember, the guys at the ventricle, they bob up and down. The cells that divide away from the ventricle, they have a different morphology. They're multipolar, and they divide in place. They don't bob up and down. So they're quite different. They don't exhibit that interkinetic nuclear migration. So you have these two populations of cells. And nowadays, we can distinguish them with simple antibodies. So you do an immunostaining. Turns out that those primary cells in the ventricular zone express a transcription factor called PAX6. The secondary guys in the subventricular zone express this factor called TBR2. It's become incredibly useful for the field. So other people that have worked on the subventricular zone, this includes Ian Smart. So very early on, he was characterizing these cells, plotting out their distribution in the mouse brain. So here's the distribution of those dividing cells. He looked at many different facets of the subventricular zone cells. And then he did this really interesting study in 1961. So he was injecting tritiated thymidine and then looking in the postnatal brain at what types of cells were being generated. And he looked at P3, P4, and also in the adult. And in P3, he saw that there were neurons being generated. And in the adult, he saw that there were also neurons being generated. So he's one of the first people to demonstrate postnatal neurogenesis. At P3, he would look, a month later, and he would still see neurons. So he was saying, these are long-lasting neurons. And in the adult animal, he would look, a month later, several months later, and he couldn't find any of the newborn neurons. So he came to the conclusion that they were degenerating. But he was just simply looking in the wrong place. So here you see plotted out, the tritiated thymidine labeled cells, and you can see them going into the olfactory bulb, but he wasn't looking there. When he looked at his sections, he was just looking radially up into the cortex, and he was looking in the wrong area cortex. So he almost, in 1961, almost demonstrated postnatal neurogenesis, but he was just looking in the wrong cortical area. I like to show this slide because it's a lesson to us all to really keep those blinders wide so that you can capture something interesting when it's right in front of your eyes.
He made this mistake because he had such a strong expectation of where it should be?
Perhaps,
also
yeah.
because the technology just made it so difficult to have a broader...
I think the technology was okay, but I think he had an expectation of where to look, so he was just looking straight up. He was imagining that they would migrate radially. So
confirmation bias.
happens to us all. So moving ahead then, in 1970s, Rakesh did a lot of wonderful work where he was looking at the radial fibers of these precursor cells, which were called radial glia by then. And what he demonstrated is that, and again, this is work in monkey, you have these palisades of the radial glial fibers coursing out, and that one of the functions of these is to serve as a migrational guide. So we saw this in Barbara's talk yesterday. This is the same figure. So there's your neuron. It'll migrate along that peel fiber. So they serve as guides. They help deploy these newborn neurons to the proper cortical area. So he had these little proliferative zones, and they would deploy cells to specific locations in the cortex. Yes.
Told me recently that the radioglial idea has been challenged. Is that, and I didn't check up on that, is that?
In which way? Which aspect
that
of
there's
it?
no longer that this idea of a neuron going along the glia has been
So,
challenged?
I can mention that in the rat. So I did a lot of time-lapse studies looking at that. The fidelity breaks down the further away you get. So in clonal analysis, about a third of the cells in the rat will stay attached to the fiber the whole way, and the other ones get further and further away, but within 5 to 10 cell bodies, so fairly close. But this is for the excitatory cells. The inhibitory cells are taking a completely different route. So your fibers are lined up this way, the interneurons are migrating tangentially to that. So this is in the rat.
But then how many cell bodies between the fibers?
How many cell bodies between?
Two of these glia fibers,
Oh, yeah, I don't know.
right? Because sometimes you say,
yeah.
well, it might move away from one, but it gets close to the other.
Yeah, and it could be, and the fiber next to it could very well be the ant of the
Exactly.
mother fiber.
That's what I'm saying.
Yeah, so,
That's not clear.
yeah, I don't think it's been investigated in a very thorough way. So another finding along the story was that in 1988, Vern Cavaniss' lab published a paper where they showed that the radioglial cells are mitotic, just like neural precursor cells. And so as of around 2000, the way the sum of the field is that you had mitotic radioglial cells, and this was visualized and identified using immuno and birth dating techniques. And then there was also some sort of neuronal precursor cell, which had not been visualized and had not been identified, but we knew it was there. It was just understood that it was there. And then there was also, it was known that you had this subventricular zone cell, and the idea was that these were all the glial precursor cells. So the idea was that you had two distinct proliferative zones.
Wait, one more question for clarification. Why were people so strongly convinced that there had to be the neural precursor cell?
It was an idea that people liked, and it just stuck.
Oh,
And
okay.
it still remains to some extent. So, and it starts right back here in 1907. So he said that there's two cell types. There's the neuronal precursor cell that's undergoing division, and then the spongioblasts, or the radioglial cells, that are a type of glia, and that those are two distinct things. And that this cell must have its own type of precursor. And that was also, in the 1980s, there was a paper where they used GFAP, and they came to the conclusion that GFAP labeled radioglia only and not neuronal precursors. And it ended up that was wrong. That was limited by technology, though. So Wilhelm His saying there's two precursor cell types, Sauer saying no, but despite Sauer saying no, people really like this idea, and as we were talking about, it still sticks around today. So we looked at this, and the approach that we used, this was some work that I did as a postdoc, was to label dividing cells with a retrovirus. So you could make an injection into the lateral ventricle, you'd have all these viral particles, they would float around, they would contact cells, and if those cells would undergo division, then GFP would be incorporated into them, and you could just look at them. If that cell that was green continued dividing, all of its daughter cells would be labeled with the GFP. So you could look at single cells or clonally related cells. And this is an example of one of those injections. It's quite easy to do, especially if you have 4 hands.
Is that a blind molar?
No, it's a manatee.
Mine's
So you do these injections, and then the GFP takes 18, 24 hours to express, dependent on the cell cycle. And if you look 24 hours later, this is one of the labeled cells. So this is one of those radioglial cells 24 hours later. If you wait 72 hours, you have a radial diploid clone. And you can see, you were questioning the fidelity. Close to the ventricle, the fidelity is pretty high. So the daughter cells remain attached to that fiber. But we're doing these experiments, and at this time we really didn't know which cell was undergoing division, who's actually generating everything. So at this time, Vimentin was considered the marker of radioglial cells. So I just did some co-immuno. You have your GFP labeled cells labeled with the retrovirus, do some co-staining, and was able to show that these cells express Vimentin. And if I looked at 72 hours later, I would have Vimentin positive precursor cells in the VZ. And in some follow-up work, we used an antibody to a phosphorylated form of vimentin. So it turns out that in M phase, the vimentin molecule becomes phosphorylated, and it labels the mitotic cells very nicely. So here's the cyto11 labels the DNA of the cell, and you can see a prophase cell here, metaphase cell, anaphase. You can see the sister chromatids being pulled apart. And in red is the phosphorylated vimentin, so it's a very punchy, it's one of the best antibodies. It's very, very nice for labeling the cells. And then did the analysis of, the simple question, what proportion of cells dividing at the ventricle express the phosphorylated vimentin? And it was 100% at all the ages. And I got that result and I thought, that can't be right. 100%, that kind of answer should scare you. So I said, I've made a mistake here. So I had another student do it, had another friend do it, and we all came up with the same answer. So surprisingly, all of the cells express vimentin, which made us think, gosh, everything's radioglial cells. Really, I think what the answer is that they're all in the same class. So they're all within the same class, all these dividing cells at the ventricle. Next, I used some physiology to try to characterize the cells. So you have these nicely labeled GFP cells, and this was some of the funnest work I ever did. So you do this retrovirus injection, and a day later, 2-3 days later, make some slices. And rather than just blindly going around and patching cells, you have a target, you're hunting it, you're going after that cell. And if you're like hunting some cell for an hour and you finally get a whole cell patch clamp on, it's just so satisfying. It was really fun. So I wish I was still doing that. That was really fun. So I included some red dye in the pipette to confirm that I've actually labeled the target cell. And there were several questions at that time. We didn't know. We could see these blobs, and we presumed they were cells, but it was possible that they could have been part of the radioglial cell. But you can see that including a dye in there, it didn't label these cells. So it labeled that peel fiber that would go out to the peel surface, but it wouldn't label the other cells in the clone. And by doing recordings from members of the clones further and further away, basically what you have is a maturational gradient. So this is a recording from one of those radioglial cells. They have comparatively speaking, a very flat curve. They have these outward voltage-gated potassium currents. And the young daughter cells, many of them will have inward voltage-gated sodium channels that you can block with TTX. And the further away you get from the ventricle, the stronger they get. So basically, as the cells are moving away, they're maturing and turning into more mature neurons. Also, by doing a retroviral injection, pulsing with BRDU, we could show that these radioglial cells are synthesizing DNA. They're mitotic. That's an indication that they are, in fact, the cell that's generating the daughter cells. But really, the best way to confirm that was to watch. So make an infection, make some slices, put your slice on a microscope, and just take pictures and watch. And you can see this is time 0. The cell's doing that interkinetic nuclear migration, comes down to the ventricle, divides, and you've got 2 daughter cells. So then the question is, what are those daughter cells? And again, just using electrophysiology, you can see in these 2 cell clones that the daughter cell has the inward voltage-gated currents, the radioglial cell would not. So again, this looks like a radioglial cell generating neurons. Now the clones could be multi-celled clones, or they could be single-cell clones. So you label with the retrovirus a dividing cell, and that GFP goes into one daughter cell or the other. It's a random decision. If it goes into a post-mitotic cell, then you end up with a single-cell clone. If it goes into a cell that continues dividing, you would have a multi-cell clone. So we would see these single-cell clones in the cortex. And one thing to keep in mind, again, is that you have these GABAergic neurons migrating in. So if you're looking at a single cell, it could be GABAergic. So just doing some immunostaining, you could see that these guys are not GABAergic, so they're presumed glutamatergic neurons. And then you could do some live cell imaging on them. So here's a case where I did an injection, day later make a slice, put the slice down in some culture conditions, and you've got your green cells, and then the fun begins. So you just put that in the microscope and you take pictures every so often. So here's one of those single cells, time 0. And they do this interesting thing where they actually orient towards the ventricle first, and then they reverse directions, reverse polarity, and start migrating away. And at this stage of development, this was starting at around E16 in the rat, it took 4 days for these neurons to make it to the cortical plate. And so they're acting like neurons, they look like neurons, but really to confirm it, what I preferred to do was, rather than use immuno was to do patch clamp recordings. You get a patch clamp recording, you're listening to the cell. I really believe that much more than I believed immuno. So again, here's watching that cell, doing this orientation toward the ventricle, turning around and migrating away. And after 3 days in culture, obtaining a whole cell recording, and you can see these inward voltage-gated currents, again, some confirmation that you're looking at a neuron. So in 2001, the conclusion was that radioglial cells are actually the neuronal precursor cell, and that the embryonic SVZ had these glial precursor cells in a holding pattern. Was that actually true? So in one experiment, I was looking at one of these cells, and the radioglial cell underwent a division. You can see the two daughter cells, and then this daughter cell away from the ventricle started blobbing up, and at first I thought it was going supernova, that it was dying and just going to explode, but it actually divided. And I said, wow, that's fascinating. And then just then, the culture got contaminated and grown over with all kinds of crap, and I didn't know what the answer was. That was frustrating. But having seen those pairs of cells being generated, then I started looking for them. And once you're aware of something, then you start seeing a lot of it. And in fact, there were pairs of these cells all over the place. And none of the pairs of cells stained with GABA. And when we obtained whole cell recordings from them, we could see the inward voltage gated currents. They would respond to glycine and GABA, all these indications that these are neurons. Oh, I should point out here too, that when you see this blob, some people were thinking, well, maybe it's a single cell, it's all part of a giant thing, but by including a dye that doesn't go through gap junctions, for example, you can see that you've got one cell labeled in that recording and the other one doesn't. But then again, using the time-lapse technique, and I'll show you 3 quick time lapses of these pairs of cells, so just looking around, I could find them all over the place. And in all of these cases, they would do the same thing. They would do this orientation toward the ventricle, and then you can see them extending fibers down towards the ventricle. And then they would reverse polarity and start migrating away. So that's the first case. This is another example. So here you're starting with 2 cells. That first movement towards the ventricle, then they begin migrating away. And I think there might be one more movie in this set, yeah. And they exhibited a number of really interesting properties. How much of this is culture artifact remains to be known, but here's an example where you have those 2 cells, and one of them starts extending processes along the ventricle, and then turns around and joins its sister cell, and the 2 of them continue migrating away. So then, again, confirmation that these are actually neurons, so they're exhibiting these migratory patterns. So in 3 cases, I watched the cells divide. Here's an example where after 75 hours of time lapse, then put them in the physiology rig, obtain whole cell recordings. Here's cell 1. You can see this is a pretty crappy recording, but you can see those voltage gated currents. Put cell 2 down. It's also got them, and those were blocked by TTX. I could wash that out and record the inward voltage gated currents again. So these are neurons. So it appeared that we had these cells, these pairs of cells, and trying to put it all together where they come from. This time lapse here will give you the answer. So I showed this movie once to a group of journalists, and one of the guys said, "This is kind of interesting, but it would be better if you showed it in real time." It's like, I'm happy to do that, but you're going to be here for 4 days. It's not going to be exciting. So here you can see the radioglial cell undergoing division. I got better at identifying the radioglial cells that were going to divide in G2 phase because the end foot on the ventricle gets fat just before they enter G2. So that really cut down on the length of the experiment. They would divide within 2 hours after I found them. So they would generate a daughter cell, and that daughter cell just gloms onto the mother. And in fact, you can't tell. At this point, you don't know which one is which. Post hoc, I could say it's this one here. But while you're watching it, no idea. They're just really glommed onto each other. But after about a day, the daughter cell's, all right, I'm mature enough, I'm moving out. And it starts migrating away. And at this time, I was thinking this is a daughter neuron. So I just wanted it to mature long enough that I could obtain a recording. And what I was actually trying to correlate there was angle of the cleavage plane with the type of daughter cells being generated. That's a whole other story I won't get into. But here you can see that the mother cell resumed that interkinetic nuclear migration, went back down to the ventricle, divided. You can see those 2 daughter cells. So I focus my attention back up here to this presumed neuron. It's migrating along, and luckily I waited long enough because all of a sudden it started plumping up. They get quite large before they divide, and that divided too. That really surprised me. And let's hurry up here. Those cells both remained, in this case, they remained attached to that peel fiber and then resumed migrating. So now I had 2 cells, and I waited another day till I thought maybe they're mature enough to get recordings, and then put them on a physiology rig. And the idea was to get a patch clamp recordings from all 4 of them to identify the cell types. And by identifying the cell types, it was basically a simple question: are you a neuron? Are you not? What type of voltage gated currents do you have? So these 2 daughter cells out here had these inward voltage gated currents that glial cells do not exhibit at all. And the daughter cells down here did not have those. And one thing, there's a couple things that we learned from that. And one of them is that this is one of these vertical cleavage plane divisions. Those are supposed to be symmetric divisions, and it turns out that they can easily be asymmetric. So one daughter cell migrates away, it generates neurons. The other one resumes interkinetic nuclear migration and divides at the ventricle. So but the most important thing is that the radioglial cells generate these intermediate progenitor cells that make the neurons. And so, putting it all together, it looks like the radioglial cells can generate neurons either directly by asymmetrically producing a neuron or indirectly via the intermediate progenitor cell. And in a lot of follow-up work, it seems like the bulk of all the neurogenesis is actually going through this process, this 2-step radioglial cell, intermediate progenitor cell, neuron.
Steve, have
Yeah.
you or anybody else transcriptionally profiled those 2 sets of cells?
No, that would be a great thing to do. Yeah, to look at, in more detail what they are.
And then mess with the machinery or not?
Yeah. Yep. No, we haven't done that. We've been, a lot of these experiments, I just consider them as defining normal, what's really going on in here, and trying to understand it. So the end result is that we have radioglial cells that are neural precursor cells, and SVZ precursor cells also generate neurons, but the radioglial cells do it via the intermediate progenitor. It seems more often. Well over 90% of the time, I think they're generating them indirectly. So what happens after neurogenesis? So there's this concept of translocating cells. The translocating cells had been identified over 100 years ago. And this is from one of my thesis talk, actually. And I had a slight interest in this as a graduate student. So we knew that radioglial cells would detach from the ventricle and migrate away. And one of the things I was interested in as a grad student is, could I interfere with the development and the timing of this translocation? And then when I started my postdoc, people were looking at these translocating cells. And one idea that was very prominent in the early 2000s was that when the radioglial cell divided, it would actually give that peel process to the neuron. And then the neuron had a very simple task. It would just translocate within that fiber that they said would become the apical dendrite. So its migration didn't need the radioglial fiber. And I didn't believe it at all until I did this experiment, and I was like, well, son of a gun, they're right, there's that cell. So then my task was, this will be easy. I've just got to patch clamp those guys and see if they're neurons. I first tried immuno, and the results were inconclusive. But in doing, I have a couple movies. I think we have time for this. So again, here's a G2 radioglial cell. You can see that fat process there. And these interesting little varicosities. And the varicosities are kind of cool because I don't know what the heck they are, but they will travel down into the cell, and oftentimes after that little packet gets consumed by the cell, then it divides. It's almost like it's a little piece of information. So again, you can see one of these divisions. And what this is going to show is the generation of a translocating cell. So the radioglia go through a terminal division at the surface, and it generates one of these translocating cells. So the 2 daughter cells, one of them maintains that fiber. And again, you can see the the radial glia and its daughter cell maintain close attachment. And in this case, rather than resuming that interkinetic nuclear migration and going back down to the ventricle, at a certain point it just breaks contact with the ventricle. You can see this process just disintegrates and it just continues moving away from the ventricle. And here the daughter cell, I don't know if it's hitchhiking on the mother or just locomoting in the same direction, but there's all sorts of interesting things going on there. So there at the end of that movie, you can see that translocating cell, and this is where they are in the slice. There's your ventricle, that's where it all started, and that's where it ended up after 4+ days in culture. And I think I have one more example. The radioglial cells exhibit a wide variety of morphologies, and these could all be different phenotypes. So they do all express Vimentin, they're in the same class, but they have a wide variety of morphologies. Some of them very simple and plainly bipolar. Other radioglia have a number of extensions that come off the cell body. And this is an example where you see a case like that. You can see these little fibers coming off here and there. It's got this strange foot. It looks like it's kind of sitting down with its leg extending there. But when it's ready to undergo M phase, it draws those processes in. This remains throughout. There's one of those varicosities coming down. Enters the cell and then it divides. And it translocates away. So what this movie is going to show is that the translocating cells remain mitotic. So they detach from the ventricle and they continue migrating away. And in order to keep on time here, I'm going to skip ahead. And when I do recordings from these translocating cells, they have more developed outward potassium currents that are much stronger. And any daughter cell that's generated, so they remain mitotic, any daughter cell that is generated after the radioglia detaches from the ventricle, none of them have neuronal properties. So it's almost like while they're attached to the ventricle, they can produce neuronal cells. Once they detach, in my hands, in my recordings, they no longer do that. Then they generate astrocytes. And previous lineage work had shown that in some cases you can have pyramidal neurons and astrocytes in the same clone. And this may be a mechanism for generating that. People really like the translocating cells, and initially there were some folks who were saying that translocating cells in a certain part of the subventricular zone, they are responsible for cortical expansion. And the idea was that despite the fact that we had published a good bit of work in 2004 and 2008 on these translocating cells, people were saying they don't exist in the rat, and that's why the rat has a lissencephalic brain, but they are in the ferret and the macaque and the human, and that's why they're gyrencephalic, but that ends up not being quite right. So actually here's a translocating cell in a mouse spinal cord. We've seen them in chicken brain now. So the translocating cells are, I think they're a common feature in developing vertebrate central nervous system. And there are people who are also saying that the SVZ and specific components of it are necessary or at least correlated with gyrencephaly. But the ferret, as we saw, has a very gyrencephalic cortex. But if you look at its subventricular zone, it's much more similar to that of a rat rather than a macaque. So this is something that really needs to be worked out. What about other vertebrates? So turtles have these cells that undergo division away from the ventricle. And you can just do a Nissl stain. So here are two mitotic cells at the surface of the ventricle in a turtle, and we can see them dividing away from the ventricle. But there's a number of people who say that the turtle does not have a subventricular zone. So what we did is we wanted to say, well, can we demonstrate TBR2 expression in the turtle? Does it exist? And it turns out that it does. So this is looking at the dorsal cortex of the turtle. And what we found, the TBR2 is in red here. Green is PCNA, that's a mitotic cell marker. And the TBR2 cells are kind of randomly distributed throughout the ventricular zone. They're not in their own discrete lamina. And what they actually look like is rat at E15. So the subventricular zone isn't generated, doesn't appear all of a sudden. So the TBR2 cells in the rat at E15, they look just like the turtle. They're randomly distributed throughout the subventricular zone. At E17, they coalesce into a distinct band above the ventricular zone. But in the turtle, we didn't see that at any age when we were looking at the dorsal cortex. However, The turtle has a different type of organization. They have this structure called the dorsal ventricular ridge, which is not laminar, but is thought to be correspond to different cortical areas in mammals. And in the DVR, we actually see that there is a subventricular zone. There's a tight band of TBR2 cells during neurogenic phases. They're mitotic, they're undergoing division. We looked at chicks. Chicks have a subventricular zone, undoubtedly. In some areas, it's very, very thick. This is in the mesothelium. We looked at dove brain. We see the same thing. So chicks and doves do have a subventricular zone. So this is a structure that's been around for quite some time. So just putting this all together, TBR2 cells have been present, and I would guess that they were present in the ancestor who gave rise to both mammals and to reptiles. So this is something that evolved very early in the nervous system. That's what the evidence suggests. Right now we're looking at crocodiles. So we've got TBR2 staining in crocodiles, and we're looking at whether or not they have a subventricular zone. So the end result of all this is that neurogenesis is a two-step process via the TBR2 cell. So your primary radioglial cell that expresses PAX6 produces that TBR2 cell, and it generates the neurons, and that'll deploy up into the cortical plate. So we talked about mechanisms for promoting growth, and really not mechanisms, but cells that are necessary in order for growth to be promoted. But when you think about it, there also has to be a flip side to that equation. So you want growth of the brain, and within each species, you want your brain to be big enough but not too big. So there also have to be mechanisms that act as brakes that restrain it. And you want a balance between the two. So you want mechanisms promoting and restraining growth so that your brain achieves the proper size. And this is a question that hasn't gotten as much attention. And so what we're looking at right now is whether or not microglia could be playing that role. And how did that happen? So I had a grad student in the lab. We got some fetal macaque tissue that was donated to the lab. I gave it to my student and I said, here's your project. Just describe every single precursor cell. It's a simple project. Just go ahead and describe them all. So he got markers for the dividing cells, figured out which ones worked in the macaque brain, and he labeled all the usual suspects, found them all. But then there was this stubborn 5% that we couldn't label with anything. So this is phosphohistone H3. And for several months, my grad student Chris was throwing on different antibodies. It got to be an expensive project, but we also were lucky enough to have many people lend us antibodies. And finally, as is always the case, the last antibody was the correct one. And they ended up being microglia. Had no idea they were there. It was a really wonderful discovery that you had these microglia in the brain. And the first picture he showed me, this is the macaque brain, was this picture, and my eyes popped out of my head. I was like, wow, the proliferative zones are just chock full of these guys. What the heck's going on? So what are microglia? They were initially described by a student of Cajal named Del Rio Ortega, and what he said back then, just by pure observation, is that they enter the brain during early development. They can have an amoeboid or rounded morphology. They often use vessels and white matter tracts as migratory guides. They're in all the different brain regions. They can be branched and ramified. In the more mature brain, they're evenly dispersed. They have a defined territory. In pathological tissue, they undergo transformation. And they can often acquire this amoeboid morphology. And they migrate, proliferate, and they phagocytose. So he came to all these conclusions just by looking at tissue. And he was spot on. This is all correct. Cajal did not like this work. He did not want him publishing it. And there's a very long story, interesting story there, but we don't have time for that today. Chris was looking at this in the macaque. And you can see some of these same cell types. This is almost a ramified cell. There's very, very few of these in the developing brain. The vast majority of them are in this range, in this middle range right here. So what does that represent? So people who work on microglia in the adult brain and pathological and normal brain, they call these rounded cells activated or cytotoxic or M1, and the cells that are ramified as resting. So they're considered to be resting, and when they encounter pathology, they draw in many of their processes. The processes get fatter. They become more amoeboid looking. And so that's called M2 or neuroprotective. It turns out that this is, limiting in some ways, but it's still a useful guide for helping you to identify different cell types. The traditional functions of the microglia is that they survey their environment. When they counteract foreign pathogens, they will eliminate them. And they will eliminate dead, dying cells, any debris they encounter. More recently, it's been found in the postnatal brain that they are involved in synapse development and also mature synapse function. So you have this idea that actually there's a quadpartite synapse. So you have your pre and post cells, pre and postsynaptic cells, astrocytes, and now microglia are thought to be playing a role in the synapses. And I pulled this off of YouTube. This is an immune cell. It's not a microglia, but it's a phagocytic cell. And here it's chasing a little bacterium. And this is what these cells do. They are highly sensitive, highly responsive, and they are relentless. They just never give up. It's just going to chase that little bacterium until it eats it, and then it's on to the next. Who else is out there? This is what they do. This is one of their chief functions, or one of their functions. So here you're looking at a paper from Davalos et al. In 2005, and they had a mouse animal in which all the microglia are labeled with GFP. They did some recordings, and then they drilled a teeny little hole in the skull, dropped an electrode down there, and put a puff of ATP. And they go nuts. The microglia go nuts. So you can see how rapidly they respond to stimuli and foreign pathogens. So the point is that these cells are highly responsive. They comprise about 5% of all cells in the adult brain, but until very recently, very little was known about microglia and their function in the prenatal brain. In fact, when my student came to me with these immunos of microglia in the brain, I ran to my computer, opened up PubMed, typed in microglia, cerebral cortex, cortical development, zero, nothing. There was no papers on that. So that made me think either we're doing something really wrong or this is very exciting. So
Do they form networks or they really stay independent?
do they form networks or stay independent? In the adult brain, when they disperse, they exhibit this tiling. So they each have their own territories.
But then they don't form connections
I don't know.
with each other.
Yeah, that I don't know.
All right.
Yeah, I haven't explored that at all. Early on, they exhibited a number of interesting characteristics. So we looked at the distribution of microglia in during development in the rat. And here you're looking at, in blue, all cells labeled, and then in green, the microglia. And you can see that there's very few of them. E17 in the rat, this is after neurogenesis has started. You can see again that in the cortical wall, there's very few of them. At E19, they start increasing in number. So rat has a gestation of about 22 days. The cortical neurogenesis occurs in the 3rd week. And so at E13, there's very few of them. 17, more of them start coming in. 19, 20, they start filling up. And so you start getting an accumulation of them, and it's bottom heavy in the rat. So they're more towards the proliferative zones where you have those neural precursor cells. We did the same thing in the monkey. One observation is that during development, we don't really see them in the cortical plate. So when they were first described, people thought that they were migrating along the pia, and you can see that there are quite a few of them, and that they would migrate down blood vessels and make their way down towards the ventricle. But if that was happening, we should see them in the cortical plate, and they're just not there. So
I think I've asked you this
Yeah.
5 times over the course of our relationship. Where are they coming from?
well, we think that a good many of them are coming in through the ventricle, through the choroid plexus. The choroid plexus is just chock full of them.
Right.
And when you look at you do a frozen section, I think it's not a good representation of how full the choroid plexus is. In some sections, the choroid plexus is huge, and it might even be touching the ventricle. So that it's like they don't even have to swim, they can just step right across. And in some sections, we see what we imagine, to represent these cells stepping from choroid plexus into the VZ. So in the monkey, again, a similar distribution. So very, very few of them in the cortical plate. So this is from what is supposed to be an E50 monkey, and you can see that there's very few of them. Here's E65, and again, neurogenesis in the monkey is from around 45 to 100, 102, something like that, in the visual cortex, as Rakesh has worked out. By 80, you see a dense band of them starting to accumulate down near the proliferative zones, and at 100, towards the end, they're chock full. So in macaque, you have gestation 165 days. The cortical neurogenesis is in the second trimester. So again, very few, and towards the end, they start filling up those proliferative zones. And in the monkey, all of the neurogenesis is occurring in something called the inner subventricular zone and the outer subventricular zone, and that's where we really see these cells. Another interesting thing, if you look at a rat, you just do some coronal sectioning from front to back and stain for microglia, you have an even distribution, not true in the primate. So here we have a single coronal section that Chris immunostained, and we're taking radial slices at different points in neighboring cortical areas, and you can see you get very different distributions of the cells. So that signifies them either responding to different cues that are present in different parts, or perhaps they're helping to contribute to areal differences. This is something that we're looking at now. The morphology of the cells also varies by location. So in both rat and monkey, in the ventricular zone, they're more likely to have more processes. In the subventricular zone, fewer. So they're responding to cues in their environment, or perhaps, contributing to differences in the environment of different locales. Similar pattern in humans. So we have a couple stages of human development, 15, 17, 20, and at 20 we start seeing an accumulation. Huge caveats in this study because we have so little tissue. We need more. What is the function? What are these cells doing? So what we did is some triple and quadruple staining using a marker from microglia, EBA1, and they're labeled in green. The primary cells that express PAX6, those are labeled in blue. The secondary guys, the TBR2, are labeled in red. And we saw them cohabiting in these same zones. So what's going on there? And so Chris looked closely at these, and what he would see is microglia reaching out and touching this. In this case, this is a TBR2 nuclei, touching it, kind of wrapping its arms around it, snuggling up next to it. Here's a really ambitious guy. He's going after two of them, and then eating it. So these TBR2 nuclei and the PAX6 nuclei would end up within these cells. So these are confocal sections. We would scan through them top to bottom and confirm that these nuclei are within them. There's a PCNA, and here you can see one that's enveloping, a microglia that's enveloping that cell. And then you would find these TBR2 and PAX6 puncta that looked like they were getting progressively smaller. So here's one microglia that appears to have digested a TBR2 cell, and then he's reaching out, touching another one. He's like, hey, what are you up to? What's going on over there? So the question we asked, you see cells reaching out and touching a precursor cell and other ones that appear to have phagocytized them. So is this incidental contact, or does the contact presage phagocytosis? Do they just walk around bumping into them, or do they touch them and say, ooh, that looks tasty? And another way of looking at that is this, a friendly hug, or is it a zombie hug? And so what Chris did is he labeled the microglia and the precursor cells and did the fun part, which is just watching. And what he found is that in 90% of the cases, when a microglial cell would touch a precursor, it would eat it within 30 minutes to 10 hours. And it was really interesting because the time varied. Here's an example where a microglial cell touched this one precursor cell, ate it within 30 minutes, kind of stepped back, touched the next one, moved in, and ate that one too. So within 2 hours, it ate 2 cells. In the rat, we can find microglia that'll have 5 or 6 nuclei within them, so they're highly phagocytic. This is another example. This one is more like a tractor beam. It appears to have sucked the nucleus up into it. It didn't appear to be moving. But in those experiments, in 90% of the cases, once the microglial cell had touched a precursor, it was like the kiss of death. And then Chris looked at interactions between the cells, just looking at a precursor cell and if it's being contacted by a microglia. At E50, it was 2 or 3%, 65, 5 to 7%, 15%. By E100, at the end of neurogenesis, it was about 33, 34%. And so it looks to us that this is one mechanism to put the brakes on cell genesis. There could be some feedback signal, something saying, we've got enough cells, the microglia move in, and they help to shut down neurogenesis. Now, when I was a student, our thought, and many people's thought, was that the neurogenesis was terminated by symmetric terminal divisions. The precursor cell would divide, you have 2 neurons, no more precursor. And that's definitely happening, but it appears that this phagocytosis is also contributing to the demise. And we looked throughout the nervous system, and it seems to be happening everywhere. So in the medial wall where the hippocampus is forming, Chris called this a bloodbath. They're just getting eaten left and right. Here you can see this one guy trying to escape, and the microglia is like, no, don't think so. Here you're looking at the ganglionic eminence where interneurons are being generated, cerebellum. This is the developing hippocampus, so you can see the 2BR2 precursor cells in the forming hippocampus, and they're flanked by these precursor cells that are picking them off. But it's not everywhere. This is not an indiscriminate thing. So I have a collaboration with a group down in Argentina, Estela Munoz, and the pineal gland starts from a proliferative neuroepithelium, just like the cortex does. And this is E16 pineal gland, and they express PAX6 and vimentin, just like radioglia in the cortex. But we saw very little. In fact, this was the only example where we had something that looked like it might be phagocytosis. We looked at all the stages of pineal gland development, and it doesn't appear to be happening there. In the adult pineal gland, different answer. So here are your microglia. They look highly activated. This is a normal pineal gland. You still have PAX6 nuclei, and they're being gobbled up left and right in the adult gland. So no phagocytosis in the developing gland, very, very little or next to nothing, but in the adult you do. And these are examples of phagocytosis. So what that tells us is that there are mechanisms for controlling that. It's just not widespread. These just aren't agents of death and they spill onto the brain and start eating everything. So there are signals that keep them at bay. When we first started seeing this, we thought, we know people talk about cell death, apoptosis, so clearly these must be dead and dying cells. So we got some different markers, including tunnel and anti-cleaved caspase 3 or CC3. And indeed, we could find cases where microglia were eating apoptotic cells, but it was a teeny part of the picture. So less than 1% of the precursor cells that were targeted were CC3 positive. They didn't stain with tunnel, they didn't stain with fluorojade, they didn't appear to be pycnotic, they appeared to be normal. So there's something else going on. They are mitotic, they are expressing PCNA, for example. And the microglia are equal opportunity phagocytes. They'll eat just about anything. Here you can see that it's consuming a nu-N nucleus, but it doesn't do that in the cortical plate. We only see this in the subventricular zone. So these are neurons that are mature enough to express nu-N, but they haven't migrated away, and they're being eaten up too. Both GABA positive, so we have nu-N, GABA positive, and nu-N, GABA negative nuclei. So they're eating a lot of different things. And as I mentioned in the rat, they're eating tons of things. So here's a microglia that's got 1, 2, 3, 4. Another one over here that's got 1, 2, 3, 4, or 5 nuclei within it. They really eat a lot. What is the life cycle of these microglia? I don't know. I don't know how many cells they can eat and remain okay about that. So one of our questions was, what if we remove them from the picture? There's a couple ways you can do that. There are people who have knockouts where they claim that they've eliminated all of them. What we did is we allowed the animal to develop normally and then remove the microglia using liposomal clogernate. So clogernate is a compound that is safe. You can eat it, and it's safe for you because it does not cross cell membranes. But if it gets into a cell, it kills it. So what these clever people did is they put the clogernate inside liposomes. And then what we did is we first put the liposomes into the media in slice cultures, and 97% of the microglia were eliminated. Then we had an E20 rat in the lab. When we got this stuff, we just injected it into the ventricle. So in one case, we injected control liposomes, which have PBS, and here are the microglia 3 days later on P1. And then in experimental conditions, we injected the clogernate into the ventricle, and it eliminated 90% of the microglia 3 days later, 90%. And then we did the simple thing. We just looked for precursor cells. So here's your control, microglia, and you can see they're highly activated. The precursor cells in red. Here's the TBR2 cells in blue. But when we eliminate them, there's a huge increase. So we decreased microglia by 90%, increased the precursor cells 2 or 3-fold. So in the absence of the microglia, you have an expansion of precursor cell number. So we're going to go through this really quickly so we can wrap up on time. I know everybody's getting hungry. At least I am. So these are immune cells, and one thing that people do in adult systems is activate them, and they use mimics of infection, either a bacterial or a viral mimic. LPS is one of the mimics you can use. You can deactivate them with a number of drugs, including some tetracyclines. So The concept is that this is modeling a maternal infection. They call it maternal immune activation. Again, you can have viruses, bacteria, different things. The mother is exposed to them. She generates an immune response. There's a spike of cytokines. They're coursing throughout her body, and they actually cross, some of them can cross the placenta and get into the fetal compartments. So LPS is purified membranes from gram-negative bacteria, and that's what we used in our experiments. So a lot of work has actually been done using LPS in humans. So here's a study where they were mimicking systemic infections in humans at the NIH. And you inject this, this LPS, and within an hour or two, you have this huge spike in temperature, huge spike in cytokines, respiration of the human subjects goes up, inspiration time goes down, so they're panting, and hypoxic, so they have less oxygen going to the blood. And here's a study design. 12 subjects were randomized and they received injections of 4 nanograms. Why am I showing you this? Because I was one of those 12 subjects. So the NIH was across the street from my grad school, and they were advertising this thing for $1,000, which at the time I was like, wow, that is a lot of money. I ended up buying a nice Cannondale bike, so I suppose it was worth it. But it was useful in several ways. One, I got the nice Cannondale, but I also got to see what it's like to be on the other side of the equation, because they itemized what the pay was, and the LPS was not, I don't know, it was like $25. The thing that they considered the most dangerous was they were doing arterial taps, because they wanted to look at your blood gases after you have this mimic of an infection and your lung function, and we had to be hooked up and everything in case we crashed so that they could save us. And I was like, oh, this is cool.
I'm gonna get my bike.
Yeah. And we had to do it twice. So one time you got LPS, the other time you got saline. I got the LPS first, and I was like, thank God. And I know next time it's not going to be so bad. But the thing we got reimbursed for the most was the arterial tap, and that hurt like hell. And I think it's good for us to put ourselves in that situation. We're working with animals. We should be cognizant of what they're putting them through. It really had an impact on me, and that's something that always stays in my mind. If I'm doing an experiment, I really want to make sure that the animals are comfortable. This is a graph that I made for that experiment, and I gave it to the researchers. For some reason, they didn't include it in the paper. I don't know why. It was the worst I've ever felt. My head just felt like it was going to explode. And then by 8 hours, I was just wiped out, very, very tired. But the headache was gone, the temperature was gone. I was freezing, and I kept on asking for more blankets, and they would take my temperature and say, are you sure you want another blanket? Yeah. So these microglia, they are immune cells, and can immune challenges engage the system? So we gave pregnant rats LPS at different times, and here's the microglia. One of the markers of an activated cell is INOS. We see an increase, a 30% increase in the number of cells that are expressing that. That's an indicator of M1 activation. And then we did a simple thing. We counted the number of precursor cells. So here's your control group, and here's your experimental group where the mother was exposed to the LPS. And there was a 20% reduction in the number of precursor cells. It didn't appear to be occurring through cell death, because there was no change in cell death, no significant change. The LPS works through the TLR4 receptor, so one idea was, well, maybe we're just doing this directly through TLR4. But the precursor cells don't express that. Cortical plate neurons do, the precursor cells don't. So if it is working through TLR4, it's an indirect pathway, and that's something that still needs to be worked out. Tetracyclines deactivate microglia, that's what people say. But long story short, we saw a 20% increase. So expose animals to doxycycline just through chow, eating the chow, and there's a transient increase in the number of precursor cells. And in addition, this is what was really shocking, is that there was a 10-fold increase in the number of new N cells in the proliferative zones. So the microglia appear not to be consuming them anymore. So it's really having an impact. You can tweak the system and alter cell numbers. What is the long-term impact? We don't know, because it's possible that the system is plastic enough that you can push it and prod it in one way or the other, but you end up with the same number of cells. And these are some preliminary results which are trending towards what we might predict if there's a long-term impact, but we need to do this very thoroughly and carefully. There are no error bars, because I don't know the answer yet. We don't know if this is significant. this could have an impact on development. And there's more and more evidence that being exposed to influenza during the first trimester has a sevenfold, there's a sevenfold increase in the probability that your child may have schizophrenia. And there are studies showing that medication-naive schizophrenic patients have thinner gray matter in the cortex. And that can result from many things, including cell numbers. So it's possible that this is a part of the equation. This is something that we're looking at, and it's part of what's driving us to figure out what's going on. So, the end result, schizophrenia could result from an interruption in prenatal brain development and have a lasting impact on brain structure. How is we don't know. That I'm going to skip. Some comparative studies. So basically the microglia are in all the vertebrates we looked at. They're in the turtle. We find them in the proliferative zones. Here's examples of precursor cells being consumed in the chick. These are TBR2 cells in the rodent, in the monkey, in the human. We're seeing this phenomena in all of the species we're looking at. What differs is the ratio. So there are more microglia per precursor cell in primates than there are in ferrets or rats, turtles. So basically, there are fewer microglia per neural precursor cell in species that have a high ratio here. Same result there. So conclusions from this long talk. Neurons are produced primarily through a two-step process. Some neurons and astrocytes are produced by the same precursor cell. Microglia appear to regulate the number of precursor cells in the brain through phagocytosis, and I have an asterisk there, and it should be a gigantic asterisk because I think they're doing many other things. This is just one part of the picture. They seem to reduce the number of precursor cells at the end of neurogenesis. This is one of the breaks for cell genesis, and this phenomena is much more prominent in primates than it is in rodents. The fetal microglia are sensitive to environmental factors, and one of the conclusions is that maternal immune responses can have an impact on cell genesis. And what I think is crucial, I mentioned these gradients of development. Yesterday, Barbara was talking about frontal to posterior gradients of developments. You have the inside-out gradient that are occurring at different times. And so external events can intersect with these developmental gradients, and that can have an impact on different CNS structures or different cortical regions and different layers. So this is something that we're investigating in the lab. To conclude, a lot of this work was done by Chris Cunningham, a grad student in the lab. He's moved on, he's doing his postdoc. Janet and Anna are working in the lab now, many other people. My wife, Veronica, has collaborated a lot on the project, and many other people, including Estella, who is doing the pineal work in Mendoza. So I'll leave it at that. Thank you very much for sitting through it.
Wonderful. So now I lost the microphone. Oh, here it is. Leah.
So it's they're phagocytosing normal, presumably normal neurons, but not all of them. Are they putting out some sort of signal?
Yeah.
Because there's that percentage that they're not
Yeah.
doing. So if you could figure out that signal, so one of the things I started thinking is this could be like a therapy for tumors or removing, stuff, tumors from brains if you can figure out what that signal is.
Right.
Because do you think there's a signal of which cells
Yeah.
they're...
Yeah, and we've looked for all of the usual suspects, the markers that people use working with microglia in adult systems or in culture conditions, and so far we don't find a correlation. So I had a slide up there briefly. Phosphatidylserine is one of them, so it's a little flag that is on the inner leaflet of the membrane, and it's raised, and that's like, come get me, I'm done, take me out of here. And we find some of the targeted cells have it and some of them don't. There could be a temporal component to the signaling that we're missing, but we don't know. That's aim 2 of the grants. We don't know. We don't have a good answer yet.
But you must have looked at some of the Gerald Chun kind of, polyploidy or different disorders of division in the cells, which isn't necessarily going to be looking as, ending up and being apoptotic in those things. Do you think that's related?
It could very well be. But as
as
far as we can tell, the cells appear normal.
But I think the ones that I think that those cells appear normal in any case, you can mark,
Mhm.
you can mark multiple DNA copies, but you can't see it in this sort of,
Yeah, that
Yeah.
could be.
yeah.
But
So-
there's, towards the end of neurogenesis, there are so many cells being
Mhm.
taken out, especially in the primate.
Mhm.
Like a third of them. So
Mhm.
one possibility is that the system just produces more precursor cells than are necessary. And one thing I would point out is that the system works differently in adult neurogenic centers. So if you look in the hippocampus, this does not happen. And it makes sense that it wouldn't, because if you take out those precursor cells, then you're done. There aren't going to be any more neurons generating. Whereas in the embryonic brain, it appears that there are far more TBR2 and PAX6 cells than are necessary, and this is one mechanism for reducing. And it's interesting to really think about it. Evolutionarily, how did this come to be?
Well, one explanation that I have heard offered is that that DNA repair occurs at non-terminal divisions, and these are all terminal divisions. So and you might even expect that to be extremely high in the biggest brains of the
It
primate.
could be.
Yeah,
Yeah,
so,
that could be. Yeah, that's a very good point.
So it seems like your brain slices are an excellent opportunity to manipulate the relationship between the microglia and the precursor cells. And in those systems, can you take microglia that you like isolate from another brain and put them on or take them off?
We're trying to do that now. So
Um,
that is one of the things that I want one of my grad students to do. So if she's successful in this, we'll get some answers. But yeah, we want to do exactly those sorts of things. They're fun experiments.
Because the nice thing about that is you can block some of them. Integrins are associated in phagocytes with the phagocytosis. In fact, they're what mediates the traction and the interaction between them. And those are things that you can manipulate pretty easily.
Yeah.
So what's the accuracy of cell division in the ventricular zone? So how much cell division leads to sort of cells that are actually not viable?
So basically, how many of them survive?
No. So you forget the microglia first.
Right. No, I know what you're
Just
saying.
accuracy
When it
of
divides,
replication. Yeah,
right.
yeah.
When it
yeah.
divides, how many of those daughter cells survive?
Yeah.
I don't know the answer to that question. There are people who believe that 50% of neurons generated are taken out through apoptosis or some other
Right.
mechanism. In order to know the answer to that, we would have to know exactly how many are produced, and I don't think we know that yet.
No,
Maybe
because
you would probably be the closest to knowing the answer to that question.
Barbara, give us a number.
I don't have a number, but but I have seen all those very nice pictures where Trina's labeled all the different versions of DNA number and DNA copies that terminal cells have, and they're just all over the place in that last division. So if there's any way of those cells being able to recognize that,
We've looked for apoptosis in normal developing brains, and in the stages we're looking at using, say, tunnel or anti-cleavage caspase 3, there's not a lot at the stages that we're looking
the
at.
point would then be, of course, that if the cell division process is not very accurate,
Right.
then you want to have your microglia in the ventricular zone to clean up the mess.
Mhm.
And this, at least in terms of correlation, this is maybe what's going on.
Right.
So
absolutely, there very well could be something that's wrong with the cell that
Mhm.
we're not picking up. We just don't have the proper label.
But would you buy that as a working hypothesis, or you think it's one of the least likely
Yeah, I
ones
think
right
that's just as viable as any other hypothesis. Yep.
So the ratio of glia to neurons is something like 3 or 4 to 1, right, in the cortex?
Depending
Roughly.
on area. Yeah.
So I'm wondering, when you're looking at these dividing cells in vitro, do you pretty much get the same ratio in that context as well?
Well, we haven't. So in vitro, we haven't really quantified the proportions of them.
Okay. Are there differences in the local environment, signaling local environment between layers, or is it all pretty much homogeneous with respect to
I think it's a safe bet that it's different.
it's different?
Yeah.
It's not known yet.
Yeah. Well, not by me.
Or he doesn't want to tell us, Mark. So, message for the young people. If the old people dominate the question period, then our future is screwed. Right? So in the lab, we have a rule, it's one brain, one question. Always by every meeting we have, every brain in the room, independent of age, has to pose one question. We're not going to impose that here, but young people rise to the challenge. Please, Benny.
This is a beautiful story, it's a very nice
talk. And it's very far from my area of expertise, so I can ask only general question. The nervous system is very organized in a very fine network of connection, feed forward, reconnected. I saw here first time that the GABAergic cells are coming from entirely different area. Do you have any insight how all these processes lead to the establishment of the very accurate and precise?
We want some answers, Stephen. Come on.
No, what is your vision?
What is my vision of how it all works? I feel like I'm focused at the cellular level, just characterizing single cells and trying to demonstrate how they operate under normal conditions, defining normal. How it all falls together it's a wonderful thing. I'll leave it at that.
But the other thing...
Yeah.
This is not fair.
I'm really
Benny,
sorry.
it's okay, Benny.
It's
It's okay.
As a scientist, we should have some goal, some big question in our.
Yeah, my question is, what are the precursor cells and how do they operate under normal conditions? And having defined that, then begin to understand how they are impacted under pathological conditions. There's a rush to knock out different genes and whatnot and come to conclusions based on a knockout. And I think to interpret that properly, we have to know normal better. So I really do consider what I do as defining normal, trying to understand it and establishing a firm foundation for how things operate in my little system. The cortex can't be a model for all, but in at least understanding how cortical neurons and glial cells are generated. And in the beginning, 4 years ago, I had this simple idea that I wanted to derive a mathematical equation, X precursor cells equals X neurons, how many do you need? But then we come to see that microglia are in there picking some of them off, so it's getting more and more complicated the more that I learn. But really, simply put, it's just defining normal. So it's not overarching, how does the whole thing
Stephen,
come together? But
I know you're speculating on the finite capacity of our working memory. But I do remember at the beginning of your talk, you declared a rather different question, which is how do we account for the expansion of the cortex? Am I right?
That's I put that up there as a very common question in the field. That's commonly asked, how does it get bigger?
Right.
And one point I want to make is we have to consider that there are influences that are impinging and restraining that growth, and we have to consider the balance. So right now we're looking at things that are acting more as a brake.
But the other thing that really struck me as something so odd and to be explained is that at the ventricular zone, the cleavage plane is vertical, and if you move away, it becomes horizontal.
Yeah. So I believe that. So I think that the orientation of the cells in the subventricular zone is determined by the peel fibers of the radioglial cells. Mm-hmm. When you have a lot of radioglial fibers, almost all the divisions are horizontal. But there are cases where they're dividing one way or another, and if you look closely, you can see that they're following the trajectory of the radioglial fibers. Mm-hmm. And then when the radioglia disappear, then the orientation in the subventricular zone is completely random. Mm-hmm.
Tony, you raised your arm. No, okay. You did raise your arm, but it meant something else. Oh, you were stretching. Okay. Okay, Leah.
And this might be really stupid, but it seems like we operate under this assumption that there's some optimal number of neurons that a given brain should have. And that seems like it would be a pretty complicated thing to achieve. Why couldn't it be something as simple as more is better, more neurons are better, but at some point more neurons aren't better. And so you can just have this really simple system where more is better, and then at some point it's not better, and microglia come on board. Mm-hmm. And you work with these two basic push-pull mechanisms that there's not an optimal number of neurons, but more is better until a certain point. Yeah. That might be completely ridiculous, but I haven't really noodled that.
Well, I believe that there are feedback mechanisms from the cortical plate neurons that signal in the ventricular zone, and that it could be as simple as through release of glutamate in a non-synaptic way. It could be having an impact on the rate of division.
Scott.
But But another way to maybe address Benny's question is if you think about the rate of expansion and how it's different in some of those species that you looked at, and then looked at different time points, when microglia cells came on board and how many there were. And that would require, I think, that you knew a little bit more about what the transcription factors in the precursor cells that made those that are generating them. Then you could sort of correlate that, because you can correlate it under conditions that then you could sort of push it one way or the other as well. And the idea being is that there's this optimal ratio based on how much expansion there is and how it has to be pared down during that development, and how that changes with species, and maybe even taking Leah's approach with extant species that are sort of very far removed evolutionarily. Have you thought about that?
Well, not really, but one thing we are looking at is, one thing that we're trying to characterize first and foremost is, we're calling them microglia. Are there subtypes? Are there different types of cells? And if they are, where are they coming from? So the work in the mouse that everybody cites is that the microglia come from the yolk sac, that the yolk sac will produce the hemopatic cells that will go into the body and the microglia, and then the microglia stay there. But it seems to me that there's probably other cell types in there. And the problem is that they express many of the same markers, so we really haven't been able to distinguish them yet. We're also looking at whether or not there are maternal cells, maternal immune cells in the fetal brains. But to try to start that, what we're doing is obtaining yolk sacs from both monkeys and rats and trying to start from step one. If they're starting with the assumption that many of them come from the yolk sac, what are our markers for these cells, and can we follow that progression into the brain?
It's a reasonable start. Got to be able to count them.
One quick one is following up on Leah's idea of the push-pull thing. So could packing density be playing a role? And if so, could metabolic, waste products like CO2, something arising from a lack of space be a signal?
A signal, yeah.
Has that ever been?
I don't think it's been tested, but not in this system. But definitely, you could, the cells could be stressed from, highly proliferative activity, and that could be, that definitely could be stressing them. Oxidative
All right.
stress. Yeah.
Stephen, on that note, thank you very much for a great talk.